Okay, let us kick off. Welcome to the first research webinar series for Q1 2026. This is 101. And welcome to all of you. Today we are being joined by folks who have registered for this webinar from across America. And fascinatingly, these are also the hotspots where science fairs, especially ISEF affiliated science fairs, are really, really popular. So not surprising to see this, also very, very heartwarming to see participation from across the country. So welcome. More folks will trickle in as we go, but we have a lot and very good content to cover. So let's dive in. So today, we are joined by two experts from Future Forward Labs. Number one, Gopi, my partner in crime, my co-founder of Future Forward. Gopi, students mentored by Gopi have been winning science fairs at regional, state, and even ISEF since 2022. Okay, so he is the brainchild behind this program that we started a few months back. And we all want, and Gopi wants to definitely spread this knowledge and STEM education far and wide. That's the reason we started this free webinar series as well. So Gopi will take us through some very critical aspects of project identification, etc. We will dive into it. Along with Gopi, we also have Dr. Katie. Dr. Katie is a PhD in microbiology and immunology, and she will dive into biology and medicine projects, especially how do you do those when you do not have access to wet labs. That's one of the most common questions we get from our students, and Dr. Katie will take that head on. Okay, so with that, let us dive in. Gopi, what are you covering today?
So my topics basically are in time for the science fair season. So I'm going to start with what is a strong topic? How do you pick one? Specifically, how do you spot weak ones? How do you decide what category to register your project in? Because everybody is getting ready right now to submit their research to local and regional science fairs. So which category do you register your project under, right? So people have a lot of stupid ideas, so to speak. So I'm trying to basically say, don't do those things. What is a testable hypothesis, and how to create a robust research plan? Because a research plan is super critical for it to be successful. So these are my top priorities for today.
Awesome. And Katie, beyond my description, what are you covering in your session today?
Today, I'm going to go through how to do biology and medicine research based on the resources that you have, some practical ideas for doing those projects, and then also the benefits to doing different types of biology and medicine focused topics.
Excellent. So let's dive in. Over to you, Gopi.
All right. So most of the times, when students come to us, right, specifically Future Forward, and even prior to Future Forward, when they came to me, it's always about ideation, right? Where do I find new ideas? They are not growing on a tree, and you cannot take new ideas from somebody else. ChatGPT cannot generate them for you, because if it does, it's already there. So it's kind of a very, very quintessential question to say how do I find these ideas? And one of the key aspects of this is look in the future work section of research papers. If you have an area of interest, let's say, for example, your area of interest is biomechanical engineering, bionic attachments. So go and look at bionic attachment papers. Science Direct has this, Archive has it, a lot of websites have papers. Go and do a Google Scholar research. Find out the people whom you're interested in, whom you want to emulate. Find out the research that they've done. Find out the research papers. Then in those research papers, in the last part of it, there's always kind of future work or the limitations of this research, or these are some open questions. It's always there. Every paper is supposed to have it. Even if you write a paper, you are supposed to have it, right? So those questions are opportunities. Now, obviously, if you go back 5 years and look at those papers and their future work, I'm pretty sure some science fair kid has presented that research in the last 5 years. So don't bother, right? So try to be more current, more recent papers, and they will basically guide you in the right direction. Now, one of the key resources that I recommend for most of my students is Archive, right? So Archive is basically a pre-published website It has very good quality research papers, and these are papers that students are currently working on. They are working on to publish it, but they've not actually published it to a journal. So they put it as a pre-publish in archive. And primarily the reason for this is because you kind of want to get your timestamp, right? Because somebody else might come with the same research and then your research becomes old because you published late. You don't want to end up in that situation. So this is kind of like, getting a token and saying like, my research is this date and I put it on archive. So because of that, my research precedes some research somebody else would have done. So this is a cool opportunity, right? You can basically sort an archive by like the most recent date, and then you can use that to understand what your research is. And then from there, look at future work on open questions. Now, when you do pick an open question, one of the key things that you need to identify is like, if I want to close that gap in research, like, future work or limitation, do I actually have the instrument necessary to do it? Like, for example, if that research is about CRISPR, do I have access to a CRISPR, right? If I don't, then like, really that research is not going to go anywhere. Second, computational modeling, right? So do I have the skills necessary in like, protein docking simulations or like in SimScale or like, any of the other software that we need to model it computationally? Third one is time and iteration, right? So this is one thing that most students and parents ask me is like, how much time do we need to spend? Like realistically, infinite, but because we don't have infinite time, let's talk about finite time, right? Good projects, really good projects, basically spend about 100 hours overall to go from ideation all the way to like, poster and presentable information. 100 hours. So you do the math, right? If you basically spend 10 hours per week, you're basically spending 10 weeks. Realistically, students spend about 5 hours per week, which means you're spending about 5 months doing this project. So that is the level of like ISEF winning projects that you need to focus on. Now, continuation projects are some of the best that I've seen, right? The reason for that is because you go through a project in one cycle, you don't win any awards, but you get a lot of judges' feedback, right? Every judge has an idea. They basically like contribute to the future work section of your project. Why didn't you do this? Now, why didn't you do that? And then there's the feedback forms that come in as well. So use all of that information that clearly guides you saying, like, I did project A, I didn't win much over here. And this is particularly useful for 9th and 10th grade. But then here are all the gaps that the judges identified, and those gaps produce my future work, and that is going to be my continuation project. And what I've seen consistently is continuation projects go very deep into the science fair cycle. They don't get ejected in the early rounds, okay? One of the core tests that you want to run for yourself while you're doing this is the why do I care test. Like, take away the fact that you are involved in this project, take away the fact that like, you enjoy this area of research. Just as an outsider, ask, why do I care? If you are not able to justify and answer that question to yourself, nobody will. In which case that project is a drop dead. Okay, so we can go on to the next slide. All right. So topic identification, like some of the examples of this future work, like, let's say, for example, that's a 2023 paper on applying EEG biomarkers for detection of early attention deficit disorder, right? This is a great paper. But in the future work section, I actually saw that EEG signals alone are actually noisy, which makes sense, right? EEG signals are noisy, but you can do noise filtering and stuff like that. But that's not necessarily the only way by which you can do things. But can you couple EEG with heart rate? Can you couple EEG with pulse rate? Can you basically couple it with other biomarker signals, which will basically give you a better value, right? And this basically means you need accessible hardware for EEG and also for heart rate monitoring. And then it clearly extends the existing research. So this will be an amazing project to go after. Now, don't take this idea and run with it. This has been done before. So it's a previous example. So let's keep it at that. Okay. Next topic identification, please. Okay, so here the second way of identifying a topic is basically fusing two fields, right? More recently, I'm seeing a lot of this happening, right? Like biology plus AI. So using machine learning models to classify plant stress from leaf images, right? So this is basically plant sciences, but then it brings machine learning to it. Music plus computer vision. Last year, one of the projects that went deep in the science fair circuit was using kind of like, computer vision to correct violin playing, right? So stuff like that. Mechanical engineering plus software. Like, you have a 3D gearbox. The problem with gearbox is like, you always react, you are always changing gears manually, but can you actually build a gearbox and simulate the load on the gearbox? And depending upon the load, you can real-time control the gear ratios so that your efficiency is always high in real time, right? So this is basically coupling control software with mechanical engineering. So when you start putting these kind of projects together, like, multiple areas, the climate science is also an amazing one, right? So urban building geometry alters wind flow. So can you simulate this using kind of like CFD? So all of that stuff is basically like, a combinational problem, and that is amazing. All right. So next slide, please. So one of the key examples is like, violin instruction, like I just hinted upon this. So you play the violin, which is great. But then like, when you're playing the violin, you kind of hear yourself and you convince yourself that you're playing it right. But early on, students are not even able to detect that they can play it right, which basically comes to the equity training problem. So is there kind of like a violin tutor, which can actually look at your hand position, which can look at your fret position, which can look at bow position, the bow speed, and where you're actually holding it, put all of that together and actually give you corrective feedback in real time to say you're playing it right or wrong. Now you can also take it multi-model. You can say it's not just computer vision, it's also going to listen to the output sound of the violin that's coming in, and then it's going to use both computer vision and sound as two signals, and that becomes like very, very high level research, right? So that's kind of like a combination of music plus computer vision. Next slide, please. So the third type of topic identification that you want to build around is basically start from a measurable gap, right? Most students that we talk to generally come like, like just yesterday I was talking to a student. They were starting on their research journey, and don't worry, it's never too late, right? It's better to start even if you have only 8 weeks than to not start at all. Right. So basically they were saying, oh, I want to do a project in fire detection, like forest fire detection. Like, why? Why do you want to do a project in forest fire detection? And if you can't answer that question, why, and you can't know like what you're going to do differently and what you're going to measure as an outcome, then that basically means you read something somewhere, you thought it was cool, and you basically want to do it. And that's it. That's basically what you're trying to do, right? And those kind of projects will always fall flat. The acid test for selecting any project is, can you point to me one metric that you're trying to optimize for in your project? And if that metric is measurable repeatedly, consistently, with whatever equipment or whatever software you have, then that is the perfect project to go after, because if you can prove it, it is true. It becomes a fact, right? So that's kind of why you want to work on the dependent variables and not focus on, I want to build like, a robot that basically can drive itself. What are you measuring, right? Next, please. Okay, so here is a cool example, right? So urban traffic plus data science. One of the weak examples of this is basically to say, oh, can I basically detect when there is traffic on the streets using computer vision? That's great. You can always detect when there's traffic on the streets using computer vision. But can you actually predict it, right? So most of the times what happens is traffic signals react after a congestion or a huge pileup happens, but they don't actually preemptively react and increase the time for traffic lights on and off for a period of time, right? So this is basically a core problem of all traffic signals. And if you talk to Department of Transportation, they will tell you that they will pay money to actually solve this problem. But there is no algorithm today or no machine learning model today that actually allows you to do it. But they have camera data for all traffic signals across all high traffic zones. You have all of that camera data, put a computer vision algorithm, start basically annotating the data saying high traffic, low traffic, high traffic by the second, by the minute, and then basically start building a machine learning algorithm that can predict when the traffic is going to go high and the traffic is going to go low. And then you can compare real data to your predicted data and see whether your actually algorithm is doing better. And you can prove not only to your science fair judges, but to Department of Transportation that your algorithm is clearly better, right? So this is a very measurable value. So always start with a measurable value. Okay, next please. Okay, so the next big piece that I wanted to cover today is category selection, because like literally every student that I'm talking to right now is basically in the process of submitting their science fair research to their regional science fairs, and they're in the process of topic selection. So some of the things that I wanted to clearly call out, if you look at the image on the top right? So this image is basically kind of called the overlap image. There's a lot more to this image because we basically analyzed every single topic against every single other, ISEF category, right? Every category against every other category. And the square box basically represents, if it is yellow, that means there's a lot of overlap. If there is dark blue or black, it means there is absolutely no overlap, right? So if you start looking at it from that viewpoint, you will see that, for example, microbiology has a lot of overlap with animal sciences, right? And similarly, if you see physics and astronomy literally has like almost no overlap with any of the sciences that are below it. Look at biomedical health sciences along with translation medicine. You will see there's a lot of overlap. Same with cellular and molecular biology with translation medicine. So projects in these areas could kind of like pivot in any one category. So category choice becomes very, very important when basically going in front of judges or like even signing up for the science fair, right? So it's the same core question, but you can look at the same project in different lenses, okay? So the key thing to remember is categories are often split by whether the contribution is new method or a new scientific insight, okay? So you got to focus on, is your scientific insight or your new method, which is the category it's more relevant to? Don't try to choose categories based on like, what feels cool, right? Please go to the next slide. Yeah. Please don't choose categories based on like, oh, previous category last year, the category that won was actually ETSD. So I'm going to basically pick a project in ETSD. Please don't do that, right? It's never like that. Second one, like, perceived easiness of category. Oh, this category has only 20 people. So let me basically just go and like, go into this category. If you're thinking it, 200 other kids are thinking exactly the same thing. So there is no easy category, right? Particularly in regional and state science fairs, ultimately the people who are selected to go to ISEF are across all categories. So just because you win in your category doesn't mean squat, right? And then similarly, like, please don't select categories on, I've seen students do this. Please don't select categories on, oh, my friend is actually competing in this. We'll be in the next table and we can basically like, chit chat during the judging. Please don't do that, right? That's the worst thing that you can do to yourself. And then like what sounds most impressive, right? So for example, I'm applying in computer science. So I basically want to do software engineering, right? Please don't do that. Your project should define your category and not necessarily like any of your preconceived notions of what a cool category is. So if you look at all of this charts in the bottom, that chart basically says on an average per competing project, how many projects actually win an award at ISEF. And this is for 2023 year, right? So we are a data-based company, so we go very deep into data. So this is all data that we have curated. So if you look at it, like, engineering, technology, statistics, and dynamics has like almost a very, very high ratio. Similarly, system software has like, a very high ratio. But that does not mean every project basically goes to system software. If you look at 2024, this will change. If you look at 2022, this will change. Okay. So every year, this kind of changes as like, the highest award winning category. So you should not do your decisions based off of this. Okay. Next slide, please. Okay, so now we're coming into the research plan part, right? So how to create a strong research plan. And the most important things is what do you do and what do you not do, right? So always start with the testable question. How does bow speed variability affect pitch stability in violin performance? Perfect, right? So if you move your bow really fast, basically, is your pitch more stable? Or if you move your bow really slow, is the pitch stable? That is a very, very clear, measurable outcome, right? And the bad hypothesis is not testable. How can I become a better violinist? Better is a very, very neutral term, right? It's a very subjective term. You don't want subjectivity in your research. You want to have objective variables that you can actually measure and prove that you're doing better. Okay, that's the first one. Second, limit the variables intentionally. I basically have students who want to do research and they want to do for 20 different input variable changes. Right? That is great. 20 different input variable changes you can do during your PhD. Okay, absolutely. Feel free to spend 5 years and do it during your PhD. KT will tell you that's what it takes. But the problem is when you are doing a science fair, you want to limit the number of variables and gain confidence that your hypothesis is indeed true. So don't try to change too many variables and don't try to take on too many variables. Limit as many constants and only focus on the very limited number of variables that you can measure. Right? Now, when you start a project, have a clear measurable endpoint. Pitch deviation, as we mentioned earlier, was a measurable endpoint. Timing error is a measurable endpoint. Bow angle is a measurable endpoint, right? So these are all very clearly measurable. There is no two interpretations of this. Sounds more musical, better player, feels smoother, feels nicer, more melodious. All of these things are very, very subjective. There is nothing you can do to prove to a judge that this is actually more melodious, right? If it's not quantifiable, it's not usable. So please don't do that. Similarly, plan for repetition and controls. If you repeat the same experiment 10 times, you should get approximately the same answer. If you do it once and go and present it in front of the judge, the first question they ask will be, what is the p-value for your test? And if the answer to that is, I don't know what p-value is, you might as well pack your bags and go home. You are not winning that competition. Okay, so statistical significance is super important. So you got to make sure that you can repeat this experiment any number of times. In fact, one of the core aspects of research is that after you present your research, somebody else who takes your presentation or your paper and repeats the same experiments with the same conditions should be able to achieve similar results. That is the very definition of a research paper, right? So if you are not able to do that yourself, then imagine how somebody else is going to do. That doesn't qualify as good research. Last one, design for analysis, not just collection, right? So when you are collecting data, you want to see several facets of the data. You want to break it down by, oh, here is basically a dimension that I'm breaking it down by. Here is another dimension. Like, for example, if you're doing a behavioral, you can say like, oh, I'm doing it by age. I'm doing it by age buckets. I'm doing it by gender. I'm doing it by time of day. I'm doing it by prior activity that they did before the actual test and the post activity that they're doing after the test. So the more dimensions you can build into your testing, the better your data will look and the more charts you will be able to put up that shows that you have done deep research. If you basically just do one dimension, you will have one table and maybe one graph. And there is no way you're going to talk for 7 minutes about that one table and one graph. And it basically shows the shallowness of the research. Okay, next one. Okay, so these are basically some gotchas that students do that we advise against. First, demos are good. They look beautiful. They look nice. They inspire you. But please don't latch on to demos as the first thing that you do, right? Building a flashy violin app is useless because the core of the research for all science fairs, for your paper presentations, for anything else, the core research is what is novel and unique. If you don't basically say that, then there is no point. The flashiness might get you like, one judge on your side, but eventually when it goes to the judging room and they're basically trying to talk about all the projects, the depth of the project will basically hold you back. You don't want to do that. Don't rely on any single trial or performer, right? So this is again, like the last time I told you, you got to take multiple tests and see whether your answers and your statistics are converging. If they don't converge, that basically means you have a problem. And you need to fix that problem. Either like, your control variables are incorrect, or basically your measurement conditions are not consistent, or you're basically doing a test which is across multiple input parameters and you're not able to even identify and control some of them. You don't want to end up in that situation. Don't mix too many variables. That's the same thing. Don't collect data without a comparison, right? So you should always have a baseline. If you don't have a baseline, first collect it. This will prove two things for you. When you collect a baseline, the first thing that will basically be available is that, okay, I have this baseline. I know how to collect this data, right? So baseline allows you two things. One is it basically gives you what your normal is, and it teaches you how to collect it. If you're not even able to collect your baseline consistently, forget like your tests, right? So you need to fix that first. Similarly, analysis cannot be an afterthought. As you're collecting data with every iteration, you need to do the analysis, because that analysis is going to tell you and hold you constant to say whether you're in the right path or not. Most of the times, if it feels like your data is jittery, it probably is, right? You want to basically run some kind of analysis, like, standard deviation is a very good analysis to run. Like anytime you collect data, run a standard deviation analysis to see like, is it even like in the ballpark? If my standard deviation is more than like 10% of my actual mean, That's a problem, right? That basically means your data is not trustworthy. So those are the kind of things that you want to consistently do and not leave it at the very end. When you collect all your data for 3 weeks with like, hundreds of participants, and then you go and see that your data doesn't mean anything. It's basically like, a sparse graph and there's nothing there. There's no correlation. That will be a very bad situation to be in. Okay, next slide. Okay, this is the most critical that most students are in at this point in time, like picking a title for the project. So for this one, instead of actually telling you how to pick a title because it's too common and too generic for everybody, I wanted to talk about some data. Again, we are a data shop, right? All of us are engineers here. We focus on data. The first one is the word novelty or novel or new. Basically, like, if it is in the title of the project, there's a 20 to 70% increase in awards, depending upon which year you look at, from 2015 to 2023, right? So this is a great piece of information. So try to incorporate it. Now, obviously, cat is out of the bag, which means if you do it, everybody else is going to do it, which means it's not going to be the same metric for the next year. So these are trends that we want to discover beforehand, before the year, and then drop them like hot potatoes after the science fair season is done, because they will not hold good for the next year, right? Similarly, cancer projects, right? Projects mentioning cancer represent about 10% of all the awards, right? But this has started dipping over the last few years. And one of the coolest ISEF stories is that there was this girl in 2013 who was a freshman, and she actually won ISEF all the way as a freshman, saying that she basically found a new cure for cancer. After that, that research was proven not legit. It was basically proven that it could not be done and it could not be replicated, and the data was not collected under controlled circumstances. But because of this, cancer got a bad rep. Like, one of the jokes even I say all the time is, for the number of times cancer has been cured in science fairs, there should be no cancer on earth, right? That's basically true. So it's a very overused terminology, but 10% of the awards are about cancer. So which means there's still an opportunity. Similarly, projects that basically state that they built a platform in 2015, they won 3 times as many awards as things that don't have platform, right? So which means that in that 1 year, that was the trend. But after that, it dipped. AI for differentiation, right? So merging 2 things, AI with something, right? So AI with chemistry, AI with biochemistry, AI with plant sciences, AI with music, AI with non-traditional fields, basically are starting to peak up, right? And those are the projects that are actually winning because there is something unique by pairing AI with something else. So that is a core area that you want to focus on. The new kid on the block, obviously, is LLMs and transformers. So if you are well-versed, this does not mean just using ChatGPT, by the way. This means understanding the transformer model, being able to break it down, doing some kind of analysis on top of LLM, seeing whether you can actually use a transformer for protein docking, stuff like that. More intricate research. If you can do this, then that will be a new frontier, and those will be really good projects that you can go on. So this is where I want to stop because I've been talking for 20 minutes straight.
Gopi, I do have one question before we transition over to Dr. Katie. Are ISEF affiliated science fairs the only options for students wanting to compete in science fairs, or is there more out there?
Okay, so I'm going to answer this question a little bit roundabout because it is nuanced, right? I have quite a few students who basically have done science fair at ISEF, but then I've taken that and presented to the specific categories in HOSA, right? HOSA also has a project presentation, right? Yeah. They've also taken what they built for science fairs and presented it at entrepreneurship competitions. They basically took the core research and built a story for a potential startup around it, and then presented that as a business model to entrepreneurship competitions, and then won those competitions, right? So it's not just the core research that you're presenting, but you're thinking about how does it apply? For example, Genius Olympiad, right? Genius Olympiad is an amazing competition, and it is meant for environmental related projects. So if your project is kind of like, you can squint and see, like, I can put an environmental spin on this, then you can basically go to Genius Olympia. There's also BioGenius Olympia. There are like a bunch of competitions which are specific to specific areas. And if you can spin a little bit of your project, so when we identify a project for our students, we try to keep it in an area where they can apply to more than just their regional science fairs. So they can apply to a bunch of stuff. And of course, the research paper and everything that goes beyond that also matters.
Yeah, super. Okay, there's an audience question. How much is expected from middle schoolers?
Actually, nothing is expected from middle schoolers, Athar. So basically, what they are trying to see as a middle schooler is, are you understanding the project that you did? Unfortunately, what I have been seeing in most science fairs recently is mom and dad do the project. Kid basically comes there, stands there as if he doesn't know anything about the project, talks one or two words, blurbs out like, some pre-taught crap, and doesn't understand the depth of the research, right? That is the thing to avoid. So if you want to talk about what to expect from middle schoolers, expect them to understand whatever stuff that they are doing. Don't expect them to understand whatever stuff that you are doing, because they will not understand it.
Super. Any other questions? And you can keep the questions going in the chat, and we will definitely get to it. Now let us transition to Dr. Katie. All right. Katie, over to you.
All right, awesome. Hi, everyone. So I'm going to talk to you a little bit today about biology and medical specific projects. So a lot of the time when you hear biology or medicine, you may immediately think people working with lab goggles, lab coats, and being at a lab bench, mixing chemicals, working with bacteria and everything. And while that is a big part of research now for college students and then graduate students and beyond, we're seeing a lot of different opportunities for students doing science fairs specifically. So students in both middle school and high school can work on multiple different projects. So we have experimental and observational biology, where you can use microbiology models, yeast, plants, and bacteria, kind of surrogates and model systems. Don't need access to a wet lab for that. You can also do nutrition, metabolism, and lifestyle experiments, and then environmental biology as well. So that's just one section that you can do. If you're focused on people specifically, you can also do medical and health focused specific topics. So like disease modeling for really, really gnarly diseases that affect a majority of the population, like diabetes and cancer, as Gopi mentioned earlier, and how you phrase that is extremely important. Another thing that is new and upcoming and really important for industry is drug mechanisms and molecular targets. So new ideas for treatments, diagnostics, and then also we can go farther than just people specific too. We can do sports science, injury prevention, work on the environmental side, and work through Genius Olympiad. And then there's also a whole other section for data-driven and then computational biology. So using DNA and RNA sequences, microbiome and health data, and then AI-driven drug discovery and simulations as well. It's really important to kind of think of this as a whole option, and you don't just have to cure cancer, as Gopi said. There's so many different options that I want to talk through, and if it's something that you are interested in, if you're a student or parents, you've heard your students get excited about a topic like this, we can kind of help create something that is manageable and that they'll be able to have access to. So you don't need a university lab to do real biology or medical research. And what really matters the most, which Gopi kind of touched on, is the quality of the question, logic of the scientific design, and then strength of the analysis. So is the student really understanding what they're knowing and how it's going to apply to real world situations? Next slide. And again, most people thinking about biology and everything too is the whole wet lab access problem and alternative solutions. So this is one of the biggest challenges that students are facing right now, and most don't have access to research facilities, expensive equipment, or specialized reagents. There's a lot of different safety regulations that can limit what can be done in schools or at home, especially when it comes to working with different pathogens, chemicals, or like regulated human samples as well. And again, you don't need access to a wet lab in order to do pseudo wet lab research or to do biology research. Just to go over some of the main restrictions right now, if your student is interested in working in kind of a wet lab scenario. As of now, middle school students are not permitted to work at a bench. So to even have the opportunity, they would have to wait normally until they're around 16 years old. They're limited to biosafety level 1 pathogens. So no Ebola, no scary pathogens or anything like that. There's a lot of regulations around what kind of bugs you can work with if you're interested in bacteria or viruses. And then if you have a high school student that wants to work in a lab, there's a lot of communication and regulation that has to go into actually getting them into the lab. So you'll have to have written permission, sometimes contracts and agreements with the principal investigator of the lab, the university or an industry lab, as well as parent or legal guardian permission. And then there'll have to be some form of official registration or contract that kind of outlines what that student is doing. So that's the restrictions that are in place right now and kind of what we have to work with. And again, some students may feel that without a lab, they're at a disadvantage, especially when they see kind of advanced projects at large competitions, but that's not the case. There's plenty of ways that we can work around this. And what judges really care about, also future employers and colleges, is again, the experimental design, controls and variables, and then data analysis and scientific reasoning. So some ways that you can kind of get around these barriers with working at a wet lab is using safe biological models. So a lot of students can use yeast as a biological model, plants, and then also simulations on the computer. You can also combine simple wet lab experience that you can start at your home and then add that with strong data analysis, reasoning, and real world impact. So a lot of what has to go into this is how you present it and the actual impact that it's going to have on people, plants, animals, the world, environment, kind of that situation. And additionally, you can also pair wet lab observations with computational validation. So bringing in all of these new tools that we have that are really important to industry, to colleges, and to future employers. It's huge having students who know how to work with these tools and work with this data that we have available to us now. So limited lab access does not limit your ability to do impactful science, and Future Forward can work with you to get you something that you're interested in that's still focused on biology and medicine. Next slide. So some general do's and don'ts for these kind of workarounds if this is what students you're interested in or parents you know your child is going to be interested in. There's some biology experiments that can realistically and safely be done at home or in local school settings. One of the most powerful tools for student projects is the use of model systems. So again, a model system is a simpler biological system that helps us understand complex biology. So again, for example, you can use yeast for studying metabolism, stress responses, and microbial growth. You can also use different plants that are commercially and publicly available. You can also look at different kind of environmental factors, things that don't have strict restrictions and regulations or privacy considerations with them. So again, some general do's. You can use food safe or household organisms. This is regulated especially for middle schoolers, so and within your science fair specifically. So if you have questions, ask us, or you can also ask the people who run your science fair. Another good do always is your PPE or your personal protective equipment. So wearing gloves, goggles, labeling everything. And then as Gopi mentioned earlier, you really want to include your controls, repeat your experiments. So the minimum for repeating science experiments would be 3 technical replicates and ideally 3 biological replicates. So that's a total of like 9 different situations as you keep going through everything. And then you also want to track data carefully, and especially for these presentations and posters and projects that students are doing, take pictures as you're working with this different equipment and really document as much as you can, because as Gopi said earlier, one of the best kind of shows that you have a good project is if you write it well enough and you study correctly enough using the scientific method that someone could read your paper and replicate it doing the exact same things that you did. So that's a test of good science. Some major don'ts for safety, privacy, health, all of those reasons that you kind of want to prevent is don't culture unknown bacteria. Not a good idea. There's lots of entertaining things out there, so you don't want to get exposed to something that can make you sick. Do not use very regulated human or animal samples unless it's within a lab, under PI, under supervision, that you have a mentor directly for a wet lab. Again, we can use those model systems, but you don't want to use like HIPAA regulated human samples. Dispose of those materials properly. So we don't want to be putting anything toxic down drains or bacteria down drains. So whatever you're using, you want to make sure that you're looking how to correctly environmentally dispose of it. And then additionally, anything that's regulated, you want to make sure you're documenting it. So do not skip your safety documentation. And then next slide. This is one of the most exciting things and exciting options that we have right now. So we can do a lot of computational projects, and there's a lot of resources that can help students get started. And one thing that I really want to emphasize is that modern biology and medicine is really driven by data and the data that we have access to. So scientists now at the bench are routinely analyzing DNA sequences, RNA sequences, and huge databases that we have access to that are just kind of sitting there waiting to be looked at and waiting to be dove into by future researchers to make sense of them. We can also look at protein structures, microbiome profiles, population health data using computers, and there's a lot of publicly available things that don't require a financial investment or going into a lab. And there's a lot of tools that we can work with this data. And if it's not publicly available data, a lot of times if you can connect the samples or the database to the researcher, there's a lot of communication where they can give you access to that data so that you can work with it for your project and kind of get additional mentorship. So some of the project options that we have are like protein drug docking simulations, disease pathway modeling, microbiome and nutrition data analysis, as well as AI prediction of drug effectiveness. And some of the public data sets that you can use, and there's so much out there that you can go through, get great sample sizes to really dive into a topic of interest that you have. You can go on NCBI, Protein Data Bank, Utapro, there's a human microbiome project, Earth Microbiome Projects, the CDC, World Health Organization, Cancer Genome Association. There's so many different free online sources to get data that is not restricted or regulated. Additionally, there's a lot of really exciting tools that are coming out. As well as AI, we also have all of these different free online tools that students can learn how to use. So like SwissDoc, Swiss ADME, Blast, the Galaxy Project, AlphaFold, Python. There's a ton of kind of pipelines to go from, okay, I'm learning how to pipette liquids into other liquids at a bench. That a machine can do that. Basically anyone could learn how to do that, but students learning how to use these computational tools and take it from idea, finding candidates to cure things or act as a diagnostic or finding biomarkers, going from idea to finding those like rational candidates that can be then used in a wet lab is a very valid, beneficial pipeline for students to learn. And that's kind of where the industry is going right now, and where a lot of wet lab researchers are putting a lot of emphasis in, because the majority of wet labs will have 15, 20 people who can pipette, but then they'll have one biostatistician who actually knows how to look at the data and the sequencing and go through all these samples. So I just want to emphasize that this is a really, really good skill for students to acquire and be mentored through this program, because it's applicable to getting into college, being computer savvy, working in grad school labs, and then also for future employers, knowing how to work with all of this data and use all of these tools is just a super essential skill that I can't emphasize enough. And lastly, it's low cost. A lot of these are free, high industry demand. And there's also something interesting that takes a lot of work, but there is a more of a patent pathway or an IP pathway if you can identify these different compounds, change them, increase their validity and use. And then it's also accessible. If you have a computer and internet, you can do a lot of different things with the data that's out there. So kind of a closing message for you guys is that biology and medicine is not limited to the laboratory. There's so many different fields that are driven by ideas, data, and problem solving. And then with the right tools and mentorship, students can really focus on any topic that they're interested in to contribute a lot of meaningful insights and help prepare for their future careers. So yeah, with that, I'm good for the next slide and any questions.
Thank you, Katie. We have a bunch of questions, so let's start from the one after Arthur asked. So question is from what if your project does not have a strong data component, like showing how mutating a protein might help cancer, but have not had any chance to test it or collect data for obvious reasons?
Okay, so I think this can be an issue that people run into a lot. I think the best way to strengthen an idea like this would be to provide multiple candidates and grade them based on why they're good and why they may work better or be more effective, and why you would want to avoid using different candidates. You can structure it and do it more in depth instead of just having one. People like comparisons. They like controls. So compare the thing that you've identified to current treatments on the market. Why is it better? Why is it working better? Why is it more effective? Why is it safer? And try and make, as Gopi said, make a computational analysis for that. So add some statistical analysis, try and dive into what's already out there. Or if you have a small sample set or a small amount of information given to you, that's okay. And that's where biology and medicine starts. You have to start with a few samples. And as long as you qualify that and you understand that, and your future direction is this needs to be studied on more bacteria, people, samples, subjects, whatever you're working on, as long as you can articulate that, that's okay. And that's really where science discovery starts.
Got it. Gopi, maybe you can take the next one, which is what year is the most important for college admissions? Is sophomore, junior placements the most important? As senior year is too late, as college admissions are already submitted? This is a question from Tiffany.
Okay, so Tiffany, the answer to your question is every year starting in 8th grade is super important for college admissions. Right. The reason for that is because you cannot wake up suddenly in 11th grade, which is what 90% of the students do, and basically say, I'm going to go to Stanford or I'm going to go to Harvard. It just doesn't work that way. Right. It is going to be a deliberate process of building the skill, building the profile, and building the tangible supporting information that goes to it, right? So we have students who start in 8th grade who build up a skill. Let's say, for example, protein docking software, right? So they basically build up that skill. Their project doesn't go anywhere, but the skill that they built up is going to let them build a better project in their 9th. And then they go a little bit further and they present at regional science fairs and local science fairs. And they also compete in HOSA. So now they have experience competing in these places. So when they go in 10th grade, they actually do a much better job in answering questions and they go farther down the train, right? So you want to basically, this is a snowball effect, right? So you start small, but then as you roll, you gather more and more snow and you become bigger and bigger. So you want to achieve the biggest piece of it in your 11th grade, right? So that's the goal. You don't want it in 12th, you want to achieve the biggest piece in the 11th grade. So you want to keep building towards it. But if you just wake up in 11th grade and say, I'm going to start building my profile now, sorry, it's too late.
Okay, related question by Karthik, but this is more for 8th grader. Is it too late for someone in middle school, 8th grade, who just learned about these 2 weeks ago, to start preparing for these science fairs this year? Is it better to focus for next year?
No, it is not better to focus for next year. The reason why I say that is because doing your research, however small, however little it is only one part of the equation. Going over there with 300 other kids at the science fair, having like strangers walk up to your table, ask you about your research, ask in-depth questions, and you are explaining to somebody you've never met before in your life about the depth of the research that you did, and you're defending the stuff and hearing from them what other ideas are, and then building that poster, and then like taking all the stuff out of there, and then like, sitting through that award ceremony. Maybe you get an award, maybe you get a consolation prize, it doesn't matter, right? That process of going through, even if it's a win or a rejection, that actually is very, very, very important. You need to do it as many times as you want, as many years as you want. So just because you don't have a project, if you focus on next year, you lose 1 year of all the other things that I mentioned over here.
Yeah, and Karthik, we will also emphasize that the regional and state science fairs are not the only places where a student's research project can be positioned. At Future Forward, we advise our students on what other competitions to also look at. So the season is just getting started. From now all the way till June and maybe even October, certain competitions run. So do not waste this year would be our advice.
Yeah.
All right. Tiffany has one more question, which is, my son is homeschooled. Are there any limitations for these science STEM competitions?
Not at all. Church schools, parochial schools, home schools, all of these are highly inclusive science fairs and competitions. There is absolutely no limitation. In fact, I would go back to this story, right? Like for my own son, the COVID year actually ended up being the best year because he was at home all through the year, so he was able to do a lot of research. So being homeschooled actually is an advantage because you get a lot of time to work on your project.
Super. Arthur has a question. What is the role of mentor in this process? When can a student openly inform judges that a mentor helped them?
So first things, you should always openly inform judges that a mentor helped you. There's nothing wrong in getting mentorship, absolutely nothing wrong, right? You don't know an area, you have an idea, you want to become an expert in that area. The goal is you start with a mentor, but the mentor is doing 10 different projects or 20 different projects with 20 different students. By the time the project is done, you should be an expert in that project and not the mentor. That is the goal, right? When you're walking into the judging room and presenting in front of the judges, the mentor will not be able to defend the project as well as you will be able to. And that is the goal. So there is nothing wrong in saying that you are mentored by Katie or mentored by Gopi. That's absolutely fine. Now, would you put our names in a research paper? Probably not, because you did all the research, you collected all the data, you basically did the analysis. We just helped you and directed you in the right way. So we did not co-do the research with you, we just guided you. So that being the case, you would acknowledge us, basically saying thank you, but you would not add us as second author on your paper, right? So that is where the difference is.
Yeah. And just to elaborate, so mentor has a role. Mentor guides you on the scientific process on how to do the project right. Mentor will not and should not do the research for you the way a parent should not be doing the research or the project for the student, right? Keep that very, very clear. That is most important to science fairs, right? That it should not be somebody else's work that a student is presenting. It is student's own work. A teacher is guiding them, a mentor is guiding them. That is absolutely acceptable. Next question is, For Catherine, when you say data analysis and scientific reasoning, are there any specific data analysis that judges typically look for? For example, any specific statistical programs like R?
I think for judges specifically, as well as anyone in the scientific field, statistics does not have to be extremely complicated. What people look for is really simple. You want to have a big enough sample size. If you don't have a big sample size, and ideally it would be greater than 30. For any general project, you want your N to be larger than 30. If you don't have that, notate it. Second would be, how many times did you replicate your experiment? Again, you want to have 3 biological, 3 technical, or just 3 technical, depending on your topic. Then you also want to have your significance. So your test group compared to your control, or your test groups between each other as well. Is there a significant difference, or is it just chance? Is it actually something that you can notate and computationally say, yes, this is significantly different? And then as well, you really want to look at your standard deviations and show them. It's okay to have some standard deviation, but if you are doing this research and you're seeing huge differences, maybe you need to reevaluate your parameters that you're running these experiments under. So it doesn't have to be really, really complicated. Those are the main factors that people are looking for. And if you want to use different programs for that, basic Excel, if you really want to go through everything, you can program with R, use GraphPad. There's a few different ones, and it really kind of depends on what project you're doing. You can make it as complicated as you want, do principal component analysis, and make some nice visual graphs and comparisons. But I think those core factors are the things that any judge and anyone in the scientific community really looks for.
Super. Milka has a question on wet lab observations. So by wet lab observations, do you mean publicly available data sets? If you are coming up with ideas that haven't been tested before, there would be no wet lab observations available.
This is a really interesting question, and I like how you phrased this. So one thing that I kind of mentioned is there's a lot of data just out there that has not been analyzed by different research groups yet. And this is a part where students really need to kind of dive in a little deeper, because data sets can be used in different ways, and you really need to understand all of the data that's collected for one data set. Maybe they have age, gender, time, diet, all of these things, and they're focused on cancer. Maybe another data set is focused on zone 2 cardio and weightlifting and what they did. And you can pull data from certain data sets to answer new research questions. So I think there's a lot of literature analysis that has to be done to create a good research question from this. And then there's also just a lot of data out there where if you're asking a microbiome question and you're looking for a antibiotic resistance cassette, that's all they looked for in a data set, but there could be so many other types of important genes that are in that data set, but they didn't ask that question, so they didn't even look for them. So it's really kind of pull and dig and try and create a good question with that. So it's kind of a workaround, a work through it, and there's a lot of reading that needs to be done to support that.
All right, and the last question we have from Tiffany is, in terms of the snowball effect that Gopi you talked about, do students typically switch projects every year, or do students dig deeper in the same project starting in 8th grade?
Okay, so this is a good question. Ideally, what will happen is you will pick an area and you will start becoming an expert in and around that area, right? For example, let's say one of my students actually is doing signal processing for musical signals, right? So basically any musical instrument, you pull different kinds of musical signals from it. I don't even know what signals he pulls. He basically has something called Carhat graphs and stuff like that. But basically he pulls in raw signals, he breaks them down. And then once he breaks them down, he's able to use those broken down signals to recast the same music like Taylor Swift, or recast the same music like, Zakir Hussain, or recast the same music as A.R. Rahman. So if basically you can decompose a music into its core elements, then you can put it back together in some other stylization. right? So that is kind of like the research he's doing. But he's focused, his skill is in breaking down and reassembling that music that he built in his 9th grade. So every year after that, he's kind of been using the core skill, but his research has been very different. That is one form of student, right? Another form of student that I have is he did a software project like AI, computer vision, animal sciences in his first year. And then second year, he said, I'm so frustrated with like software engineering and programming. I don't want to do this anymore. So he basically did a CFD project, computational fluid dynamics, like nothing related to AI, nothing related to software engineering, right? So students do both, and that's absolutely fine. But when you start building a core expertise, it makes sense to build around that core expertise. Like, for example, if you spend time, like Catherine was saying, right, you spend time basically doing and learning the protein docking software, which means you spent a lot of time doing it. So there's no point throwing it away the next year. Try to build a project that's going to utilize that and then gain some additional skills in order. Okay.
All right. I have one last question, and this is to you, Katie. Are you the only biology and medicine related mentor with Future Forward Labs?
So I am not the only mentor for biology and medicine. We have a few other great mentors here, and whatever topic your student is working on, we can match you with the best mentor based on our specialties. So we have Clark Roberts here. He is a neuroscience specialist and focuses on psychology, neurophysiology, complexity sciences, AI, statistics, and computational modeling. He's a PhD from Cambridge and has also worked with MIT and NYU. So he's another person, neurology specific related. And then we also have Dr. Delia. She has a PhD from Michigan, and her areas of expertise are with biomedical sciences, optometry, applied physics, and she has a lot of really cool, interesting work if you want to work with her for like eye-brain connections, neurodegenerative diseases. So we have a good mix of people that can help based on the topic that your student is interested in and the area of specialty that they need kind of an expert support with.
Super. Thank you, Katie. Folks, any last questions while we wrap up? This was our 101, the first webinar. The second one is in about a month, Research 201, in which we will dive deeper into failure analysis as your data starts coming in for experiments. How do you do that, right? Interpreting your experiment results, so on and so forth. And from a topic and subject perspective, the way we did for bio and medicine, we will deep dive into AI, ML, computer vision, and training data challenges. So that's what is coming up on Feb 15th, exactly same time, 5 p.m., around 5 p.m. Pacific is when we will meet you. And we will hang around here for a few more minutes if you have any more questions. But thank you for joining and hope this was useful to all.